In this video, we're going to talk about biological molecules. These are molecules of life. Biological molecules are divided in 4 different categories: carbohydrates, proteins, nucleic acids, which include DNA and RNA, and lipids. All of these molecules are considered organic molecules. So what is the definition of organic molecule in biology, in science? It doesn't mean the type of fertilizer you use. Basically the definition depends on what atoms the molecule have. So for a molecule to be considered organic, it has to include carbon and at least one hydrogen atom. Now all life biological molecules are carbon-based. Carbon is the backbone of biological molecules. So that's the carbon component, but having carbon isn't enough to be considered an organic molecule. There has to be at least one hydrogen atoms. Inorganic molecules could have either carbon or hydrogen or have none of them. So let's take a look at examples. Molecule number 1 is glucose. We can see that there's plenty of carbon and also hydrogen. So this would be an organic molecule. Carbon dioxide has carbon, but it has no hydrogen. So it is organic. Sodium chloride has neither carbon nor hydrogen. So it's inorganic. Water has hydrogen, but no carbon. So it is inorganic. This molecule is an amino acid. It has both carbon and hydrogen, so it is organic. Now let's talk about carbon, the properties of carbon. So as I mentioned, the backbone of biological molecules is carbon. We are all carbon-based entities. Now carbon can form a maximum of 4 bonds. And you can see 1, 2, 3, 4. And when we write the formula for molecules that are made of carbon, we usually write them in a planar form. So for example, for this, we usually write something like that. And then we write the hydrogens, and it kind of gives the impression that this molecule is sitting in a one-dimensional flat surface. But the reality is, when we look at this is a molecule of methane. When we look at this molecule, we'll see that actually these bonds are in a different space when you look at it in 3-dimension space. This hydrogen is pointing up. The central one is representing carbon. This hydrogen is pointing to one direction on the same plane as this hydrogen, and then this hydrogen is sticking out of the page, and that hydrogen is sticking into the page from the back. So this molecule is not flat, and just the fact that the bonds that carbon makes with different molecules take a different direction in 3-dimensional space, it gives it more properties, more interesting properties. You can make various different variations of molecules by just moving around whatever is connected to carbon in space. And you will see what I mean when I show you examples. So we mentioned that carbon-based bonds have 3-dimensional, that they have a shape in 3-dimensional space. They're not flat molecules. So methane in this case, ethane, there's 2 carbon molecules with 6 hydrogens. You can see that these bonds are sticking out, they're sticking to forward, they're in the same plane, so there's space in dimensional space. But if you end up making a double bond between the 2 carbon molecules, or between carbon with another molecule, then all of a sudden the molecule becomes flat. So all the bonds are in the same plane, if you like. So just not only you can make different types of carbon-based molecules depending on where you position these bonds, but you can also make carbon-based molecules flat. Now, just being a molecule that's sitting on the two-dimensional space of flat, it doesn't mean that it cannot have interesting variations, which this is an example. So this is an example of two different fatty acids, and there is a double bond in here between these two carbons. And these white bulbs that you see in here, these are hydrogens. Now compare this molecule, this is almost a straight line, but this one is bent. And this bend is very, very important as you will learn later. But what is the difference that is making? What is it causing this molecule to be straight and for this to be bent? Well, it's the position of these hydrogens. If we go back in here, we see that you have these carbons and you have molecules attached to the carbons. And if you were to draw a line from the center of this molecule, you have two sides. You have the up side here, and you have down. You can position atoms that are attached to the carbon either on the same side or on opposite sides and cause variation. So in this case, iliadic acid, you can see the hydrogens are on the two sides to the opposite sides of this double bond. This is called a trans configuration. Trans means different. So the hydrogen atoms are on the two sides of this. Remember, we said we make a plane of this plane. Oleic acid, you can see these hydrogens are on the same side, and this causes the molecule to bend. So we say this molecule has a cis configuration around this double bond. And if you've ever heard of trans fats and cis fats and the fact that these trans fats are good for you, that's why I mentioned that these are important, but I'll show you another example as well. So although this molecules of carbon that have double bonds may seem like they're boring and planar, but then you change the atoms attached to the carbon on either side of the double bond, you get interesting properties. Which brings me to the topic of isomers. Now, on each row, I'm showing a different type of isomer. An isomer basically means different forms of the same molecule. So both butane, let's stick with A for now. With both butane and isobutane, if you were going to write the molecular formula of these two molecules, they would be exactly the same, C4H10. But you can arrange these bonds, these carbon-carbon bonds differently in that in isobutane you can chain them. You can create subchains. So you can maybe take this last one and replace this hydrogen with this CH3 and then move that hydrogen here. So this is called branching. These are called when you move around the molecules, when you move around the atoms in the backbone. This is called the backbone, this chain of carbons. When you move around the carbon atoms in the backbone and form these subchains, this branching, you say you've created structural isomers. And these two molecules, again, have exactly the same number of carbons and hydrogens. The molecular formula is the same, but they have completely different properties. Then we go to geometrical isomers, which relates to that cis and trans configuration that I just explained. So geometric isomers usually refer to the arrangement of bonds around a double bond. See, this is a double bond. So in the cis configuration, you see that these hydrogen atoms are on the same side of this plane, and then the hydrogens in the trans configuration are on this different side of this plane. Then we have enantiomers, which gives you different molecules with different properties solely based on where you position the different atoms that are attached to carbon in three-dimensional space. So for these two molecules, the molecular formula is the same. In the L isomer and D isomer, you're really not moving anything around. You're just changing the position of the bonds in that three-dimensional space, such that you create two molecules that are mirror images of one another. And these are called enantiomers. Now, this is a very important concept because a lot of what happens in biology is like molecules fitting together like a lock and key. And if molecules don't have the proper orientation in three-dimensional space, things don't happen. So the L and D isomer, they may look like they are the same molecules and should have similar properties or identical properties, but they don't when it comes to fitting into different molecules, fitting into proteins and enzymes, and coming with other molecules that form a complete fit in three-dimensional space. So enantiomers make a big difference, different enantiomers, and a lot of times drugs that block the function of some molecules in our bodies tend to be, some of them are enantiomers. So basically you create the same molecule but in the mirror image of it. So just a little bit more on enantiomers to make it more clear. Just like your left hand and right hand have the same components, but if you compare them, you can see that they're mirror images of one another. And a very important molecule that makes life possible, which is the building block for our proteins, are amino acids. And amino acids can exist in two different enantiomeric forms: L amino acid and D amino acid. Now, these are enantiomers. They are mirror images of one another. So if you would put an mirror in here, one would be the reflection of the other. Now, in nature, in our bodies, the only type of amino acid that can function, that work with our enzymes and systems in our body, is the L amino acid and not the D amino acid. So the amino acids in nature are this enantiomer, not that one. So if you were to, let's say, take protein supplements that would have D amino acids, you wouldn't be doing yourself any favor because the amino acids won't be incorporated in your proteins. Okay, so now we switch to the topic of functional groups. We just finished talking about all sorts of different ways that we can make carbon-based molecules interesting and give them different properties in arranging the bond, carbons around the bond, in changing the position of these atoms, in forming enantiomers. But there's also additional ways we can give carbon-based molecules important chemical properties, and that is by the use of functional groups. And functional groups, as the name implies, provide a new function to the carbon-based molecule that has these groups. And here's a number of functional groups that are important for us in this course. So let's go through them one by one. So we're looking at a hydroxyl functional group. You need to know, just let me stop and say that you need to know the name of these functional groups and their chemical structure and their properties. So this is a very important table. Okay, so to illustrate how these molecules, to illustrate how these functional groups, which is basically a group of atoms attached to one another, so you can think of them as a Lego unit, let's see how they add additional properties to carbon-based molecules. So let's start with ethane. It's a plain old carbon and hydrogen molecule. It's a hydrocarbon. Now let's see if we can replace this hydrogen with a hydroxyl functional group. Now you notice this R here, this represents the carbon-based molecule to which these functional groups are attached. So if I were to use a different color, let's see. So then this R is the carbon-based molecule to which the functional group is attached, and it's actually this part that is the functional group. So in here, R is the carbon-based molecule to which the functional group is attached, and this is the functional group. This is the functional group. Okay, so I hope that makes it clear. So let's get rid of these now. All right. So now let's imagine that we're going to replace this hydrogen with a hydroxyl group. So now what do we have? We go back, we have hydrogen. What do we know about this bond? It's a carbon and hydrogen bond. Their electronegativity is similar, so this is not a polar bond. Now replace that with a polar bond. We know that the oxygen-hydrogen bond is polar. Let me get back my pen. So we know this bond is polar, so we have a slight positive charge here, and we have a slight negative charge in here. So all of a sudden you go from a molecule that has no polarity to a molecule that has polarity in here. And this is enough to allow this molecule to start interacting with water. So that's how the hydroxyl groups would add new functions to a hydrocarbon molecule that otherwise would just be very unpolar and would not interact with aqueous-based water molecules, aqueous-based solutions. But you replace that hydrogen with this hydroxyl group, now all of a sudden there is a possibility to start interacting with water or form hydrogen bonds with other molecules. Now let's focus on this methyl group. So we're going to replace this hydrogen with another methyl. What's happening? Is this functional group polar? No, it's not. It's just like the rest of the molecule. So this is a non-polar functional group that makes this molecule we started with even non-polar. It's more less water-loving. It's more hydrophobic. So in this case, we're really just making this molecule longer and more hydrophobic. But if you had a carbon-based molecule that was really hydrophilic, and then you replace one of the atoms that is attached to carbon with this, all of a sudden you make it more hydrophobic. Okay, now let's go to carbonyl. Now carbonyl is a functional group, so this is the functional group. This is what we're talking about. and you see R and R prime because this functional group is sitting in the middle of a chain. So then let's go down here. We're looking down here right now. So we're splitting this and we're inserting a carbonyl functional group here. And again, we have a polar bond in here. Oxygen has a partial negative charge, and carbon here has a partial positive charge, because oxygen is a lot more electronegative than carbon. So therefore, this functional group makes this molecule polar. So now let's move to carboxyl. So let's take a look. So this is the part of the functional group. That's the functional group. Let's read about it. It's charged. Well, the way it's written here is not charged. You may be wondering, well, why does it say it's charged? Because as soon as you put a molecule that has a carboxyl functional group in it, this hydrogen comes off. So this functional group ionizes in aqueous solutions and releases positively charged hydrogen ions. And since they can release hydrogen ions, they are considered acidic. So when you add this molecule, this functional group, to a carbon-based molecule, not only you make it polar, but it also you add acidic properties to that molecule. So if we replace this hydrogen with a carboxyl group, we have this bond in here, which is going to dissociate the bond between hydrogen and oxygen. So then you end up having actually a negatively charged molecule in here. So this is actually going to look like this. So all of a sudden you have a molecule that on one end, it's not only polar, it's actually charged on one end. And it also makes the solution acidic. Before that, when this wasn't here and there was a hydrogen, this molecule wasn't acidic, it wasn't polar, but you replace that hydrogen with this functional group, all of a sudden you add these properties to this boring old hydrocarbon. Now we can go to the next functional group, which is the amino. Now again, amino, it's one nitrogen with two hydrogens. That's the functional group. So again, we see that it says it's charged. Why? Because as soon as you put a molecule that has this functional group in here, nitrogen is going to take up a positively charged hydrogen ion from the solution. And that's because nitrogen has this pair of electrons sitting here that it really wants to share. It has a tendency to share these electrons. So therefore, you add this functional group instead of this hydrogen, then what happens as soon as you put it inside a solution, this nitrogen is going to want to give its two electrons to a hydrogen molecule and form an additional bond. Now, since the positively charged hydrogen ion doesn't bring an electron with it, there is an electron deficit. So there's going to be a positive charge on these molecules. So not only by adding this amino group, you're adding polarity to this molecule, because these are also polar bonds, but you also give this molecule a positive charge. So you went from a boring old hydrocarbon to a molecule that has all these interesting properties. So then we're going to go to phosphate, which when we talk about nucleic acids and phospholipids, you're going to see a lot of these phosphates. So the phosphate functional group is here. And again, you will see that it says it is charged. So when this functional group, molecule with this functional group placed in liquid water, this phosphate group also will release these hydrogen ions into the solution. So then you're going to lose these hydrogens. These hydrogens are lost. into the solution. They're lost. And then what happens? This becomes negatively charged. So again, by replacing a boring old hydrogen with this phosphate functional group, you not only make the molecule polar, but you also give it acidic properties and you make the molecule charged. Now, last but not least is this sulfhydryl functional group, which is pretty much similar to a hydroxyl functional group, except we're replacing this oxygen with a sulfur. And again, there is a large difference between electronegativities. There's a partial positive charge and there's a partial negative charge. Therefore, it makes the molecule to which it is attached polar. So I hope this explanation helps you understand the importance of functional groups and how they work to give hydrocarbon molecules or organic molecules interesting properties, because this always seems to be a point of struggle for students. And if you have any more questions, don't hesitate to reach out. Now, what do these functional groups look like in real life? Molecules, let's take a look at 3 of them. So this is stearic acid. This is a long hydrocarbon molecule with this functional group, hydroxyl functional group at the end of it. So this is what's called a fatty acid. And we're going to see fatty acids again, and the fact that you have these functional groups in here, it allows this fatty acid to form bond with glycerol and give you fat, but you will see that later. So here it is, that's the functional group that is attached to this long hydrocarbon chain, which in that previous table we would call an R. The second example is an amino acid. It's amino acid glycine. Amino acids are made up of a carbon attached to a hydrogen, and then they are attached to a carboxyl group. That's one functional group. And then amino group. All amino acids have these 2 functional groups. And then in this case, in terms of glycine, the fourth component is hydrogen, but this varies, and we'll get to that when we get to amino acids. And that's one of the reasons that we call this an amino acid. Here's the amine, and that's the acid. In this case, we call this a fatty acid chain because this part of the molecule is fatty, this part of it is acidic. Finally, we're looking at a molecule of glucose, and then we see all these hydroxyl functional groups. Carbohydrates have a lot of hydroxyl functional groups on them, and that's what makes them so soluble in water. So just wanted to show you another example of functional groups in action. This may seem a little complicated, but don't worry, these are 2. This is your DNA. That's your structure of DNA, and we're zooming on it. And we'll talk about these more later, but what I wanted to point out is these functional groups that you saw in that paper, they're all busy forming hydrogen bonds with one another. Here's an amino group, that's a carbonyl, that's another carbonyl. So these functional groups play very, very important roles in life. And in this case, they are making possible for the strands of DNA to be held together. So we're talking about biological molecules, but what are the reactions that allow life to put biological molecules together? So we're going to talk about conceptually how this happens. Let's talk about chemical reactions first. Well, chemical reactions allow us to make new molecules from older molecules. So a chemical reaction involves breaking old bonds and making new ones. So in this case, this is a reaction for burning methane. This is a methane molecule. We need oxygen to burn methane. So these are the reactants. This is what we start with. And then this arrow shows the process of the reaction. So that means this is what's going to happen. And then these are the products. That's what you get. This is reactants is what you start with. Products are what you end up with. So what do we get as a result of burning methane? We get CO2 and water. And these are called the products. So now let's take a look at the reactions that make biological macromolecules. Now those biological molecules that I showed you, proteins, DNA, lipids, nucleic acids, most of them are these really huge polymers, those really long molecules that form by attaching individual units together end over end. Imagine making a really long Lego piece, and then you take those individual Lego pieces and attaching them together. So those individual pieces that are attached together are called monomers, and each type of biological molecule has its own particular monomer, which we'll talk about later. So what is the reaction that helps life, allows life to make those big molecules like proteins, nucleic acids, carbohydrates? The reaction is called a dehydration reaction. So what do they call it? Dehydration reaction? It's called dehydration synthesis or dehydration reaction. So what's happening is that a water molecule is lost. Now this sometimes causes pause for students and say they think, okay, where was the water molecule? They look for a water molecule here to be lost. Well, we are actually, through this reaction, the water molecule is produced. That's one of the products of this reaction. And how do you produce the water molecule? You take a hydroxyl from one monomer and a hydrogen from another monomer, add them together and produce water. So this water is a byproduct of this reaction. And then we get this longer molecule that is attached together through this covalent bond. Okay, so that's two monomers coming together. Now that we have two of them, we call this a dimer. That means two. But it doesn't stop there, because most biological molecules are really, really long. So the reaction keeps going. You get a dimer, add another monomer, you do a dehydration synthesis reaction, and then you get what's called a polymer. This is a polymer. So most biological molecules are polymers. So synthesis of protein, nucleic acid, carbohydrates, they are all done by putting together individual units, individual molecules, the monomers, removing one hydroxyl from one and a hydrogen from another monomer, removing the water, and then making a long molecule. Now, in life, we not only need to make large biological molecules to incorporate into our muscles, our DNA, etc., but we need to also break biological molecules. Why? Well, because we eat. We eat protein, we eat carbohydrates, so on and so forth. And in order to take that protein that we ate and use its components to make proteins for our own bodies, we need to break that meat that we eat or the protein that we get from plants. They need to be broken down into their individual Lego units so that we can take it and build our own protein. So how does that happen? What is the reaction that makes that possible? Well, it's the opposite of what you just learned. So imagine this is a big piece of meat that you ate. It goes through your digestive system. Individual monomers are going to be absorbed by your body to help make your own muscle. So how does that happen? It's the opposite of dehydration synthesis. Last time we made water. This time we're going to add a water molecule. So remember, we stole a hydroxyl functional group and a hydrogen from two monomers. We're just going to give it back to them. So this is called the hydrolysis reaction. We're using a water and we're breaking the bond. And again, this happens over and over and over again, and we keep breaking all these bonds so that we can have individual units to build our own muscle. That's what I'm using protein as an example, but this applies to all biological molecules. So let's take a look at example. These are two glucose molecules, so we're looking at carbohydrates right now. And when we make, let's say, glycogen in our bodies, when potatoes make starch that we later on eat, how does the potato make starch? How does our body's livers make glycogen? How do we do that? Well, you take individual glucose molecules and then it undergoes a dehydration synthesis to give you a dimer. So what is happening? Again, we're removing a hydroxyl functional group from one monomer and a hydrogen from another monomer. We're going to remove them and put them together. This is a byproduct, and that's what we want. This is a dimer. Okay? This is a dimer. So that's dehydration synthesis, and this happens in nature all the time. And then that plant that makes the starch that we eat keeps doing this over and over again until you get a really long stretches of glucose molecules attached to one another in the form of starch. Now let's take a look at how we break that starch in our bodies. We have that starch molecule, or even you can have sugar, which is a dimer of two different monomers. So we have this dimer. Let's say we take this and we eat it in this form. So how do we break it down into its individual units so we can use these individual glucose molecules to make our stuff in our own bodies? Well, it's a hydrolysis reaction. It's the opposite of this one. So we're adding a water molecule, and then we're giving back these hydroxyl and hydrogens that we took from these monomers. So here's an example of really what's happening in nature of a dehydration synthesis, which is the reaction that is used to make large biological molecules, and hydrolysis, which is a reaction that allows us to break large biological molecules.